All right, good afternoon, everyone. Thank you so much for joining, and I'm so excited to talk to you guys today about the work we've been doing on conservation genomics and disease ecology of the Texas tortoise. So before I get too deep into this, I wanted to give you all a little bit of an introduction to the other folks that have been hugely instrumental to this project and are not presenting today, but especially Dr. Kasumi and Dr. Dolby led the efforts on annotating the genome and the population genomic work, along with grad students that worked with them. Griffin, Jade, Raul, and Sarah were all a huge part of this project. So just want to make sure you all saw their faces since they're not presenting this information, but they were a huge part of it. So first I want to get into, why do we even protect the Texas tortoise? Aside from it being arguably, in my opinion, the cutest reptile in South Texas, why do we care about this species? Well, for one, they are excellent at dispersing the seeds of prickly pears. They tend to favor eating them over anything else that's available in the environment, and after they pass through their digestive tract, it can help to fertilize the seeds so more of it can grow. So especially in South Texas, we have a lot of what's known as wood rats that come around, and they'll use the base of these prickly pears as support for structures they create known as middens. So that's pictured in the middle of this image. It'll look kind of like a giant stick pile, but so many different other animals, insects, other rodents, lizards, even Texas tortoises will use it as a shelter during, cooler temperatures to keep warm or just from predators. But then predators like rattlesnakes will hang around them to find things that are emerging from these middens. So Texas tortoises are kind of part of this micro ecosystem, and they create some of the support structure. And along with that, they're of course part of the food chain. So this is just one of the reasons why we want to protect our iconic Texas tortoise. So where are they found? The map all the way on the right, the area highlighted in green, represents their historic range throughout the state of Texas and into Mexico as well. The other two images kind of are representative of some of the main habitat types they're found in. So the most iconic of them is Tamaulipan thornscrub, which is characterized by a lot of, acacia, mesquite, just general brush that wants to stab you when you crawl through it to look for tortoises. But they'll also be found in more grasslands as well, especially when there's a matrix of this Tamaulipan thorn scrub and grasslands meeting. They'll go out into the grasslands to forage and then go back to the thorn scrub for shelter. But they utilize both of these, and those are the two most iconic. So looking at the map, you might be thinking, well, it's a lot of area, so a lot of places to study them. But despite them covering a lot of these areas, the bulk of the research done on Texas tortoises has focused in the lower Rio Grande Valley, particularly in Cameron County, but also Willacy and Hidalgo, and then at Chaparral Wildlife Management Area. So, if you're looking into their research, most of the studies have been done there. Now, there have been other pockets where research has been done, but the bulk is in these areas, which kind of leaves questions about, the fringes of their range and what's going on there, since most of our knowledge comes from a subset of where they actually are present. So getting into the threats facing tortoises, one of the largest ones is habitat loss, usually from development for industrial, commercial, residential activities going on that kind of displace Texas tortoises and reduce available habitat to them. Roads are another big one, can separate two patches of habitat from each other. And of course, a slower moving animal like a tortoise doesn't always have the best chance of moving successfully across a busy highway. But another one facing them is invasive grasses, especially things like guinea and buffalo grass can displace their native forage and change thermal conditions for them, so it can be a problem. But the main two we're going to be focused on are disease and the illegal ownership of Texas tortoises. Okay, so getting a little bit into a roadmap, I'm going to be talking about the disease in Texas tortoises. Chris is going to get into population genomics, and then we'll talk about our repatriation event. So first I'll get into mycoplasmosis. this is one of the diseases that has been on the radar of all of the different biologists studying tortoises because of its prominence in that it's a precursor for upper respiratory tract disease, which has been linked to mortality events in gopher tortoises and Mojave desert tortoises. So this disease picture here shows a gopher tortoise that has the nasal discharge that can be characteristic of this disease. And because of the mass mortality events that have occurred, it's led TPWD to want to know, how prevalent is it in Texas tortoises? Does it pose this sort of threat that we need to be worried about? So mycoplasmosis is caused by two different bacterium, as far as we currently know, Mycoplasma agassizii and Mycoplasma testudineum. And when a tortoise is showing clinical signs of this disease, it's characterized by nasal and ocular discharge, some swelling of the eyelids. It looks like the tortoise has a cold essentially because it's impacting the respiratory tract. And it's spread via contact of an individual that is spreading the bacteria or shedding it into the environment, and one that has not been exposed before. Once a tortoise has been exposed to this bacteria, its immune system doesn't react, so it's a chronic disease and it has it for life. So there will be times where you'll see a tortoise and it doesn't look like it's harboring the bacteria, and it might not be shedding it at that time, but oftentimes in response to a stressor like drought years, then there might be the immune system might start to react more, and it might start to shed that bacteria into the environment, have mucus in response to an acute stressor. So currently what we know about this disease in Texas tortoises is that one of the interesting things is that Mycoplasma testudineum is the bacteria that's been noted to be more prevalent, at least compared to the other four tortoises found in the United States. So that's what this bar chart on the left side is showing, right? The gray area shows Mycoplasma agassizii. That was the first bacteria that was described to be linked to this condition. And then Mycoplasma testudineum is the dashed lines. Areas black represent co-infection, where both of these bacteria were found present in the tortoises. And so Gopherus berlandieri is the scientific name of the Texas tortoise. It's the third one. You can see it kind of looks like the odd one out. Now alongside that, you'll see that range map again, and those areas circled in red represent where this has been studied in Texas tortoises. So still a bit of a limited area compared to the whole range that they occupy, even within the state. So part of the reason we want to know where this pathogen already exists in the environment is because, as we mentioned, there's a lot of development. And so when there's development, TPWD will sometimes try and work with developers so that Texas tortoises can be moved off-site. That way there's not that loss. I know most of the time they try and move them as close as they possibly can to habitat patches, but that's not always an option. So there's been explorations into whether translocation is a potential for this. And so when you're doing translocations, tortoises are going to be a bit disoriented. So normally when a tortoise is moving, it has what's known as a home range. And so they'll move in shorter steps. They kind of know their neighborhood, they know their food patches, so they're not going to move too far. They might only move, within 100 meters from where they started in a day and then settle back down. But a tortoise that's been moved somewhere new is going to be disoriented. They might be trying to do what's known as homing behavior, figure out where the best food patch is. So they'll move sometimes a kilometer over a very short period of time, which can have implications for the spread of pathogens. So the dots at the bottom of the slide represent an experiment where they were looking at how many tortoises were in contact with each other when you had a control population, and then you looked at a resident population where translocated individuals were introduced. And so those lines between the circles represent what's known as a contact event, where tortoises were in contact with each other long enough to spread a pathogen. So time 0, there's no introduced tortoises. You see there's not that much contact between the population. T1, where they've added in the translocated individuals, the population is suddenly a lot more connected. A few weeks later, most of the tortoises have now come into contact with each other. And then it eventually settles out. But in that initial period where they come in, if there's a pathogen existing in the resident population or the translocated population, a pathogen can be very quickly mobilized, potentially through the entire population. So it's something that you need to be mindful of, and already understanding what diseases are prevalent is a way to kind of think about these decisions. So the objectives for the disease side were to map where these two bacteria were prevalent in the wild, and then assess if there were any sort of predictors to the exposure status of the individual tortoises. So to do this, I did a mixture of visual encounter surveys, going to state parks, private properties, walking along game trails and whatnot to find tortoises out moving. Where there were roads available to cruise, like county roads or on private properties, we'd also cruise those during hours of activity. And then we had a really great biologist network where folks who already had permits to handle these Texas tortoises, if they saw one, they would give me a call, hang on to it, and I would get to them within a couple of hours to collect the sample I need. So I had biologists from these different organizations that were hugely helpful to increasing the sample area for this study. So once I had a tortoise in hand, the primary thing I needed to get from the tortoises was a blood sample. So I was taking as little as I possibly could to still get all the information I needed. So once I had the blood from the individuals, I would immediately stick it into a centrifuge I had in the field. That way we could separate the plasma and serum, which was used for an ELISA assay. This essentially let you know if the tortoise was developing antibodies to either of these pathogens. And we chose to use ELISAs because, as I mentioned before, this disease is chronic. So once they're exposed to this bacteria, they don't necessarily clear from it. And so late stages of infection, the ELISA can be accurate to know if they've ever been exposed before, since they might not necessarily be shedding them, which PCRs would detect, but they might have the antibodies. And this ELISA would give you either positive, yes, they are creating antibodies, negative, they're not producing them, or suspect, which means it's elevated and there's likely an exposure history. And University of Florida was conducting these analyses for us. Then the red blood cells were kept for the population genomic analysis, which was done at Yale, and all of the subsequent actual analysis part of the sequencing was done by University of Alabama and Arizona State, which Chris will tell you more about in just a little bit. So getting into the predictors I was examining, we were doing general morphometric measurements. So we would measure how large they are, weigh them, then measure their carapace length, carapace width, as well as age them to a general age class, sex them. And then I would also do a health examination at that point where I would see if they had any of those clinical signs of mycoplasmosis. So look for any sort of oddities to the eyes or nose or any previous signs that they had been going through any of that nasal discharge. Alongside that, I'm not going to get too into the weeds on the models, but I was looking into some sort of anthropogenic stressors that may be indicative of exposure status. So how many roads there were, what the traffic was like, what the population size was like at the nearest population center, and their exposure to oil and gas activity. All right, so now moving into the results. So this map here is displaying the empty circles represent tortoises that were tested and did not come up as suspect or positive for Mycoplasma agassizii antibodies at all, whereas centers where there is a blue circle with increasing light blue to darker blue represent how many individuals there came up with a exposure history to Mycoplasma agassizii. So with Mycoplasma agassizii, there was a little bit of clustering across the landscape. It wasn't just spread randomly across the area, but there was a little bit of a clustering a bit in the lower Rio Grande Valley, and then kind of along the west and a little bit the coastal bend. So there was a little bit of clustering, but it's not necessarily a trend that entirely made any sense. But I just wanted to point out those areas. And so overall, 23% of the individuals tested did show some exposure to Mycoplasma agassizii, and 6% of those individual, or 6% of the overall individuals that also had Mycoplasma agassizii did show clinical signs. So then for Mycoplasma testudineum, it was a higher percentage of exposure. About 45% of them had been exposed to this bacterium. And the spread was much more random. There wasn't any sign of clustering. Most areas that we tested, there did appear to be some exposure history to Mycoplasma testudineum, other than a few random counties here and there, and most of the counties we did not detect them in. We had only tested, less than 3 individuals there. So it could be if we tested more individuals in those counties, it may show that Mycoplasma testudineum, those tortoises have been exposed to that bacterium. But again, still only a small percentage had clinical signs at the time of testing. So again, only 5 individuals did have any sort of clinical signs at the time of testing. So I'm not going to get into the weeds on this either, but the main thing I wanted to point out when we looked at any sort of potential predictors of exposure status was for Mycoplasma agassizii. There was some slight trends when we looked at both the distance to oil and gas wells and the number of roads within a certain buffer. So as a tortoise was closer to an oil and gas well, there was a very slightly higher probability of exposure. And then as there were more roads within an area, there was also a very slight increasing chance of exposure to Mycoplasma agassizii. But the R-squared values, which can kind of help you understand how much of the variance these predictors explain, were both quite low. So there's certainly other things that better explain what's going on here that the models I ran did not capture. For Mycoplasma testudineum, it appeared that traffic on the nearest road can very slightly increase the chance that individual had been exposed to this bacterium, and it did seem that males were less likely to be exposed. But again, R-squared value was only 0.12, so it didn't explain that much of the variation, and we did test a whole lot more males. So, take this all with a grain of salt. There's definitely other variables that I think would be useful to look at to better understand what's going on with these trends. The main thing I want to point out is that the trend from that earlier study I pointed out does appear to hold looking at a range-wide scale instead of those little pockets. So I recreated that graphic from before and added our updated information. So essentially you do see that Mycoplasma testudineum does appear to be more prevalent in the wild looking range-wide as compared to the other tortoises in the United States. And again, higher than Mycoplasma agassizii. But I also wanted to bring back out that the clinical signs were very rarely detected, and Mycoplasma testudineum does seem to be mostly across the entire range. So I think long-term studies would be beneficial to fully understand the threat that it poses, but it does seem like it's likely been in these Texas tortoises for quite some time. And with that, I will turn it over to Chris to go ahead and talk about the genetics side of things.
Great, thank you, Jackie. Just these. Hi everyone. My name is Chris Robinson, and I work with Dr. Kenra Kasumi at Arizona State University. Today, I'll be talking about how we can use genomic resources to identify population structure in the native and charismatic Texas tortoise. Southern Texas, home of the Texas tortoise, is characterized by several unique ecoregions. For example, we have the Southern Texas Plains, which stretches from the edges of my home in the hill country into the subtropical regions of the lower Rio Grande Valley. It's full of plains with thorny scrubs, mesquite trees, open grasslands, and prickly pear cacti. We have the East Central Texas Plains, which typically has open sandy prairies and oak-dominated woodlands. And finally, we have the Western Gulf Coastal Plains, which stretches along the coast from southwestern Louisiana into northeastern Mexico. These are often described as tall grass prairies, but the plant diversity increases as soil salinity decreases inland. One common factor among all of these is they are split by major rivers and highways, which creates barriers for species inhabiting similar habitats. There is also subtle yet important environmental variation across the region. Across the range, we see a 6 to 8 degrees Celsius difference in average annual temperature, with this increasing towards the southwest. Similarly, the temperature in the warmest month increases southwesterly. These hotter regions of the state tend to also have the lowest annual rainfall, often exceeding or barely exceeding 250 millimeters in a year, with some parts of the year receiving little, if any, rainfall. These environmental differences offer opportunities for animals within a species to become locally adapted and diversify across the range. We can see an example of this in the humble green anole lizard. Often, you'll see these in your gardens, in areas with high rates of irrigation, such as a university, or even at your local pet store. They're found in far South Texas all the way up into Oklahoma, which offers ample opportunity for local adaptation. For example, the minimal daily temperature experienced by these animals in the winter decreases as you head north towards Oklahoma. Notice that the estimates here stop in 2014. If you think back to 2014, there was a significant weather event, a polar vortex, that significantly dropped temperatures across the southern United States. Before the polar vortex, southern populations in Brownsville were significantly more sensitive to the cold than populations in Austin, but these differences were lost after the vortex killed off many of the southern anoles. If we look across populations from Brownsville to Ogden, Oklahoma, we can see that only the southernmost populations were affected by this, suggesting that the populations were locally adapted to their environment. If we examine the genes that regulate how a lizard responds to the cold, we can see that some of these population level responses are driven by how the genes are expressed, suggesting that these responses are heritable and under selection. Finally, we can zoom in and look for single nucleotide polymorphisms, or areas of DNA that differ by a single base pair across individuals. We can see that certain genes that regulate nervous system function, and therefore how muscles might respond to the cold, differ before and after the polar vortex. Therefore, we can conclude that investigating genomic differences can help us draw conclusions about adaptation across populations. While these genomic tools are useful for inferring causes and effects of evolutionary processes, they can be fairly limiting. One requirement of this kind of analysis is sequencing the whole genome for many individuals. Even as the cost of sequencing continues to decrease, these monetary values quickly add up and could put strain on budget-conscious labs and state agencies. For example, the Texas Tortoise genome is approximately 2,200 megabases, and at the current price, whole genome sequencing at reasonable genomic coverage easily eclipses $500 per individual. Working to develop cheaper tools is critical for wide-scale application of genomic tools for conservation efforts. Despite this cost prohibition, the utility of whole genome sequencing for conservation efforts cannot be overstated. From whole genomes, we can estimate population genetic estimates that are useful in defining and refining conservation units, which are subpopulations specifically identified for conservation concerns. Here we can see the defined and redefined conservation units for the Mojave Desert tortoise, which is a close relative of our Texas tortoise. A powerful output of whole genome sequencing analyses is a variant call format file. These files contain all of the single nucleotide polymorphisms, or SNPs, found within a genome. Simply, SNPs are base pair substitutions that occur within a subset of a population. For example, we can see here in our reference genome, which is the top DNA sequence, that we have a G or a guanine in this position. However, in another individual that we're comparing to the reference, the same position has a C or a cytosine in its place. By aggregating all of the SNPs within a genome, it's possible to infer where an individual is from based on SNP similarity with other individuals. Given all of this, we wanted to see if we could apply these concepts to our local Texas tortoise. While it is illegal to collect these tortoises, people often do in hopes of improving their lives, even while they're already adapted to their environments. As individuals from the Texas Parks and Wildlife Department recover these animals, it's a logical question to ask, where did this tortoise come from? Often it's not easy to get this information, so we tested whether we could develop an efficient and cheap assay using genomic data so we could confidently conclude that a tortoise came from, say, the southern Texas plains and not another ecoregion. To do this, Jackie from Texas A&M at Corpus Christi traversed southern Texas and collected small blood samples from each Texas tortoise she came across. For each of these samples, we extracted DNA and conducted whole genome sequencing, averaging a bit over 10x coverage, allowing us to accurately detect any variants in the genome across the range. Griffin McDaniels from the University of Alabama at Birmingham then took these genomes and performed variant calling analyses, which uses high-performance computing clusters to identify variants within the genome, or SNPs, with high confidence. If we reduce the dimensionality of these data, which allows us to examine the major axes of variation rather than having to comb through hundreds of thousands of SNPs independently, we can see that genomic variation is correlated with latitude or along a north to south gradient. Similarly, we see that genomic variation is also correlated with longitude or along a west to east gradient. Finally, if we just examine the first two major axes of variation in our genome, or those two axes correlated with latitude and longitude, we see that there seems to be three non-distinct clusters of individuals that can be identified by genomic variants. Collectively, this suggests to us that we can use these variants to identify approximately where a tortoise is from, since the information contained within the genome varies across space. I won't get into this too much, but if we use computational algorithms that approximate how many groups of individuals there are within a population, we see equal support for 1, 2, and 3 groups. This is nice because it confirms that all of these individuals are from one species, yet suggests there is enough variation in the genomes to create population subsets, which may be useful for identifying sites of origin. To explore these subclusters a little more in depth, we used discriminant analysis of principal components. This approach similarly identified 3 populations from the single nucleotide polymorphisms, but the genetic differentiation among these populations was modest at best, with all of the FST values, which is an estimate of genomic differentiation, being close to 0. This, once again, suggests that while there are subpopulational differences among these individuals, they all belong to the same species. However, if we examine just the leading 10,000 SNPs of the first loading from the model, which should define the axis by which the most variants exist, we start to see significant differentiation, with these values eclipsing 0.2. This suggests to us that just a small subset of polymorphisms, 10,000 or less, could be useful in identifying genomic variants that are related to latitude and longitude. So our next step was to see where these populations were and if they segregated spatially. Here again is a map that shows the 66 tortoise samples collected by Jackie, where each point represents an individual. If we color these points based on the subpopulations identified by our former analysis, we can see there are 3 distinct clusters of individuals. Population 1 is in the eastern part of the range, population 2 is in the southern part of the range, and population 3 is in the western part of the range. This was a really exciting result for us and suggested that we are onto something important. If we map the rivers and roads onto this map, we can see that I-35, which goes from San Antonio down to Laredo, and is a major source of import-export traffic, largely defines the boundaries between populations 1 and 3. And that Highway 359 to 285 here defines the boundaries between population 1 and 2. There are a few anomalies, of course, but we'll discuss those in a minute. We then needed to test if, using only these 10,000 or so polymorphic sites, could we predict where a tortoise was from? To do this, we used a machine learning algorithm called Locator, which takes known location information to train a model and then predicts locations of unknown individuals from these training sets. We masked the location of each individual iteratively and then performed this analysis for each individual to see where the model would predict it was from based on similarity with known individuals. And what we can see is the model performs pretty well. So here we have open circles that represent the predicted location and a line that that represents the real known location of that predicted individual. On average, a tortoise's predicted location is only 67 kilometers, which is about 40 miles from its actual location. Further, there is movement towards central location of each subpopulation. The model didn't know about these populations, but still identified the same substructural population. Overall, this suggests to us that these few genomic variants can be useful in identifying the location of origin of a confiscated tortoise. Quickly, we will look at a few specific ones that appear to be anomalies. We have a Western individual, identified from all models to be part of population 1. We have a cluster down here that appears to have individuals from populations 2 and 3, and we have a cluster over here that appears to have populations 1 and 2. The interesting thing about these is that they're all municipalities. This is in Eagle Pass, this is in Laredo, and this is in Corpus Christi. While we can't know for sure, these may represent animals that were translocated by humans and released back into the wild. For the next steps of this work, we're partnering with Tangled Bank Conservation, who uses genetic tools to support wildlife and conservation projects to develop assays so we can quickly genotype these SNPs. For example, Tangled Bank has identified population structure and cryptic speciation in green salamanders, watershed level structure across alligator snapping turtles, and isolation by distance effects in gopher tortoises. They offer several genetic techniques, but here we will use next generation sequencing, specifically a process known as dd-rad-seq, to sequence our pre-identified polymorphisms. Fortunately, this method requires only a small amount of blood to obtain genetic sequences and costs less than $150 per individual. Then finally, we have to figure out how and if these methods are actually useful for conservation efforts of Texas tortoises by applying them to samples we collect from confiscated animals. Back in August, the Texas Parks and Wildlife Department, in collaboration with our friends at Texas A&M Corpus Christi, held an event in Laredo where we see 2 subpopulations of tortoises represented, where people could give up their illegally owned Texas tortoise, no questions asked. This event was shockingly successful, with over 150 tortoises surrendered within 2 hours of the event's start. Sean will talk more about this, but we are well on our way to having proof of concept for these applications. While we hope these efforts are impactful for conserving Texas tortoise populations, we'd be remiss if we didn't acknowledge the difficulties in repatriation. First, we know there is adaptation to different habitats, so imprecision in our estimates could put a tortoise in the wrong area if it matters. Second, as Jackie discussed, we have to be cognizant of diseases and how it could spread among naive populations. And finally, the climate in Texas is changing at an alarming rate, and soon the adaptations that we are striving to account for may not be well suited for a given habitat much longer. Conservation biology aims to strike a delicate balance between the past and the future, so all we can do is do our best. And with that, I'd like to thank you for your attention. And if you have any questions, feel free to reach out to me at my email here, and I will turn it over to Sean now.
Good afternoon, everyone. I'm Sean McCracken. First, I want to thank Jackie and Chris for that presentation, for their presentations. And I'm going to talk a bit kind of briefly about the next, part of gathering or taking all of this information that we've learned from the genomics and our disease studies and actually applying that towards conservation efforts of the Texas tortoise. During the course of this project, we became more and more aware of, different individuals from different agencies, confiscating Texas tortoises, like the Border Patrol, or game wardens finding, someone with many pet Texas tortoises. Online spaces where people were talking and showing their pet Texas tortoises. So we realized that this issue of people keeping Texas tortoises as pets was probably much bigger issue than what was initially thought. Jackie, I have to say too, handled a lot of these calls about these tortoises that had been confiscated or reported by whoever that discovered them. So our next project was to develop this Texas tortoise forfeiture and repatriation program that Chris alluded to a minute ago. And the idea was to have an amnesty forfeiture events. The original plan was to have many of these events where people, no questions asked, could come and drop off their pet tortoises, and we would then use those tortoises to test repatriating them or rewilding them back to the landscape. In early August, as Chris mentioned, is when we had our first event. Yes, within the first little over 2 hours, 155 tortoises had been turned in. Even before that, leading up to the event, 30 tortoises had already been turned in to the location where we had the event. And I'd like to mention too, we only did like us and a number of our partners basically did one social media post 2 weeks before the event to advertise it. And this was the response that we got from that event. So repatriation, right? This is just returning an organism to its place of origin. You often hear organisms referred to as being, a waif tortoise, for example. And these are neglected or abandoned individuals or individuals that have been, kept in captivity or removed from the wild. Now, a repatriation program has been shown to be successful in gopher tortoises. And so this is what we wanted to try to attempt with these pet tortoises that had been forfeited to us. So this is where we kind of took all the projects that have been done, our work along with others' work, combining them together to actually, employ this project of forfeiture and repatriation. So we had the knowledge of, as Chris explained, where do these tortoises come from? We could test them and identify, within a little over 60 kilometers on average, where that individual came from. We are able to identify the risks. We run a disease panel we work with University of Florida as well as University of Illinois to look at a panel of 24 diseases, because we don't want to be introducing novel diseases to naive populations. And then we had an idea of how long we actually needed to keep Texas tortoises in what we call soft release pens or enclosures. So most turtles and tortoises, have this homing behavior or homing instinct. You know, if you pick up a Texas tortoise and move it to a different location, it's going to try to find home. And so by putting them in these soft release pens, we can basically break that homing instinct, and the hope is that they learn where they've been placed in this pen that that is their new home. And some work that had been done on a different project by folks at TAMU Kingsville and the Caesar Kleberg Institute had identified, they tested up to 12 months. Our plan is to actually test between 12 and 18 months. When we had this, you know, when we put this plan together for these events, we had to put together what we refer to as our quarantine network. So this is partners, amazing partners who really stepped up for this, that would be able to take the tortoises that had been forfeited to us and hold on to them while we did all of the testing and screening that needed to be done before we could put these tortoises back out on the landscape into these soft release pens. And I forgot to mention, you know, when we planned to have this first event in Laredo, we expected 30 to 40 tortoises, and that was going to be a successful event to us. You know, we basically got 6 times that number. And I don't remember if I mentioned it previously either. I think I did, but I just want to repeat that we have about 100 tortoises on a waiting list as well, in addition to those 180. So huge shout out to all of our partners that were able to quarantine tortoises for us while we took all of this previous project information and processed it and applied it to these tortoises that were collected. And so within just the past couple of weeks, we finished building our soft release pens. We now have 11 hectares of pens built in various locations. It's silt fence that's buried 6 to 8 inches deep, so they can't get under it, and hopefully predators won't dig under it either. And it's all circled by electric fence as well. So electric fence low to prevent burrowing underneath, and then a higher electric fence line, hopefully to prevent anyone from jumping over inside of our pens. To date, we've released about 130 of those tortoises that were forfeited. All of the hatchlings, so there was a lot of hatchlings and very small juveniles. Those are being kept separate and overwintered where they can be closely monitored. I know people probably have a lot of questions about the forfeiture event. That is something that we'll talk more about in the future when we do another one of these webinars on that particular project. That is an ongoing project, like we said. Hopefully it's going to get cold here sometime soon, and those that we have released into pens will go into brumation for the winter. They all have tracking devices on them. They've all been pit tagged, so we'll be going and checking on them periodically to see what they're up to. Huge shout out to all of our funders, all of those who have given property access to us. I want to point out there's a lot of individual private landowners that have made their properties available to us. Jackie thanked the Biologist Network, and then a whole bunch of data assistants as well. So thank you to everyone. I want to thank Jackie and Chris and all of our partners working on these research projects. It couldn't be done without all of y'all. I got to back up and thank Jackie, because she's the one as a postdoc who's really been heading up this forfeiture and repatriation project. And as you can imagine, getting 6 times the number of tortoises that you expected has been a daunting challenge, to say the least. And with that, we have a whole bunch of field assistants to thank as well, too many to name. Again, this has been a huge group effort. And with that, we can all, I think we're ready to take questions.
Yeah, well, thank you very much, Jackie, Chris, and Sean. The questions are starting to come in, so that means everything's working okay. Sometimes the chat can be weird, so please post your questions in the chat, and I'll read them out. Okay, they're starting to come in. So first question: you said there were no questions asked at the surrender event, but did any of the people volunteer how and where they obtained their tortoises?
Yeah, so I should say we did ask a few questions. We did ask everybody, you know, if they were willing to answer how long they had had the tortoises, where they picked them up from, and if they'd been kept with any other reptiles. That particularly to know if they were any sort of crazy kind of unexpected pathogens we should be aware of. You know, if someone's keeping them with like a monitor lizard or whatever, it's more important to know that. But yeah, most people did, and most of them had picked up tortoises from within Webb County, where Laredo is. So most of them were not from a very far distance, which I think is to be often expected when you're living somewhere where there are tortoises. Most people just picked them off the road as they were traveling somewhere.
Have you investigated whether the genetics can explain any of the disease occurrences?
So I know that's been it hasn't been done yet, but that is has been a topic of conversation among all of us as future steps into looking into yeah the genetics and how that interplays with the disease. Sure. So not yet, but that is in discussion.
Next question is, what is the status of the 60+ tortoises that were removed from the area in 2022 around the port of Brownsville to make way for new LNG facilities? I don't know. Wasn't part of this project, but I think we can kind of address that a little bit.
Sure. So those were held, they kind of staged different release timeframes. They did something a very similar setup to what we've done with creating these soft release pens, and they have now removed all of their fencing material, and those tortoises have been allowed to disperse from those areas. Again, they tested up to 12 months time, and 12 months seems to be sufficient in general for breaking that homing instinct. But not in all cases did it show that did it prevent kind of, straight line movement once those fences were lifted. I am not aware of that was a couple of years ago now. I am not aware of what the current status is of those tortoises from that project. And I'm not even sure that they're still being tracked, but yeah.
I don't think they are, but there will be some updated information being published about that in the next, 3 to 12 months. So yeah. So these tortoises were released in one of the 3 zones that their genomes matched? Question mark.
They were released nearby. Yeah, they were released near. So since they generally all came, we had a mix of some from like Bexar County, Webb County, and some were released in Webb County, but they were also released in a larger property, just a couple counties over. So within the same habitat type for sure.
And we have multiple properties where we've built these pens. As you hopefully understand, we just can't divulge where those locations are.
Will there be more forfeiture events in the future?
It is something we are hoping to do more of in the future.
I guess I'll just add to that yeah, we're worried about, we were overwhelmed at this event, right? As you can imagine. And we're worried, and this was in Laredo. So our concern is, what would an event like this result in, if we were to do it in Brownsville or McAllen or Corpus Christi or San Antonio, like these larger population centers? We just fear we're going to end up with like 1,000 tortoises. So we're trying to devise a plan that maybe doesn't require actual events where people come and drop off their tortoises, but it's some sort of reservation system to drop off your tortoise. I don't know if you have anything to add to that, Jack.
No, I was gonna say we're trying to find a way to do it in a more controlled manner, so that way it's not just, we can get a number that we're expecting as much as we can. Oh, okay, there's Dr. Hankey.
Oh, good.
So for the question about the status of the tortoises in the chat, Dr. Scott Hankey, who is leading that project, left to address that.
So can you remind us how long the tortoises in this project will be kept in the soft release pens?
So a year is the current plan, especially since they were released in the fall. The thought is that since it's fall time, tortoises are getting settled to brumate. So even when we take the fencing away, both the 12-month period and the time of year, since it's fall, they shouldn't be moving as much as, say, in the spring.
Awesome. Oh yeah, I see Scott's got a comment, and I'm sure everybody can see that. Question here said, I'd be interested to see the difference in dispersion patterns in forfeited tortoises that have already been released versus the hatchlings that were overwintered. Great talk.
I think something to follow up on that a little bit too that will be interesting to see is we had people who brought in basically multiple generations of tortoises. They had adults, they had sub-adults that were offspring of those adults. There were hatchlings that were, probably a mix from different individuals, that they had as pets. One thing we didn't mention too, as far as the events go, this was kind of traumatic for some people dropping off their tortoises. So we are hesitant as well to kind of, we want to make it an easier situation for folks in the future as well when they're dropping off pets that they've had for 20 plus years, right? These are long-lived animals that, right, can outlive us. So, with most tortoises, they are long-lived, and it's almost like, a parrot or a macaw that you have to will, in a sense, or figure out who's going to take care of that animal that's going to outlive you.
Yeah, that's a great point. Another question here, is every tortoise released being tested for a large array of potential diseases and being genetically tested for probable origin before release?
So before release, just given the volume we got, we were not able to test every single individual, but we did make sure to get about half of every. So if a single one was forfeited, we tested that one, but from each group we made sure to get, I think we ended up with about half of every single group, so we could get a solid representation of what each group was made up of. And the disease panel tested for over 26 different pathogens. We also included some wild tortoises in that testing as well, since we had primarily been focused on Mycoplasma, to ensure that any pathogens we were releasing kind of matched what was known in either the same area or very nearby to where we were releasing. So disease was definitely our primary, and we did meet with like the TPWD vet when we were coming to this decision of where we felt comfortable. And we are going to be following up with some more testing too, since they are still in soft release pens, if things need to be changed in any way.
I guess I'll follow up a little bit on that just to clarify. So like I said, we had people who turned in, multiple tortoises. So for example, if someone turned in 10 tortoises that were all living together, we tested like 5 out of those 10.
Got it, great. Comment here that San Antonio will be a large forfeiture area too. Great presentation, great presentation. Okay, somebody, Might have missed this. Do you see any evidence of inbreeding or outbreeding depression in conjunction with habitat fragmentation in these populations? And are these factors considered when repatriating wave tortoises?
So when we look at the population genetics, we don't really have enough individuals to break it up into fragmented populations. Overall, we see an average heterozygosity of like 0.25, so not super high inbreeding. It's about what you might expect for wild reptile populations. As we get more of these individuals in and do more sequencing, we probably can consider habitat fragmentation, but right now we just don't have enough data to be able to confidently conclude whether I-35, for example, is fragmenting these closely related tortoises in any meaningful way.
That's great. Excellent presentation. Good hopes for the future and important field application of rewilding science. Great presentation. So that does appear to be the end of the questions. Yeah, I'll close out here. Jackie, Chris, Sean, thank you so much for your time today, for coming to talk to the group. It's great work, and I'm sure everybody's going to be on the edge of their seats to see how the forfeiture stuff. So we'll definitely have you back in, in a couple of years, I guess, to update everybody.
Thank you.